Hi everyone, I'm Ho-Yin, and today I'll be presenting my dissertation topic on orchids, and the research title is Can Plant Communities and Soil Factors Predict the Establishment of Helleborine Species on Lindisfarne? So Lindisfarne, where is it? It's towards the Northumberland coast. My specific area that I'm studying in is the Snook, and that's just slightly before the island itself along the causeway. And here's the study area. It's actually quite large. Lindisfarne has numerous site designations from National Nature Reserve to Ramsar and Special Protected Area and a Site of Special Scientific Interest. And that's largely due to its habitat supporting numerous species like the dark green fritillary, birds such as ring plover, and its botanical interests such as sea thrift and orchid species like the early marsh orchid. But today we are looking at the Marsh Helleborine as one of the orchids that I'm going to study. Well, I did study. It's widely distributed within the UK, but populations are sort of scattered. And in the northeast, Lindisfarne is probably one of its strongholds, and it's usually located within the moist dune slack habitat. The other orchid, the Lindisfarne Helleborine, was first discovered in 1958. It sort of undergone a lot of taxonomical name changes. It's sort of like a pasta parcel. Everyone's sort of unraveling a bit of its identity. It was formerly known as Epipactis sancta as its own species. However, in 2007, through some DNA sequencing, it was placed as a subspecies of Dune Helleborine, also known as Epipactis dunensis. In the northeast, there's another Helleborine that you might get confused with, and that's a variety of the Dune Helleborine known as the Tyne Helleborine. And this Helleborine can be found near Riverside or Calaminary in Grafton, but really to separate them apart, knowing its locality is sort of the best way to separate them apart, because if you're on Lindisfarne and you see this, it's the Lindisfarne Helleborine. You can't get it mixed up. So why did I pick these two orchids? But they both occupy different areas within the dune system. This is a cross-section of a dune system that you may find on Lindisfarne. The Marsh Helleborine, as mentioned before, is found within the dune slack system. It's created by blowout from wind, and it makes a depression in the sand dunes, leaving the sand very close to the water table, as you can see through this diagram. The Lindisfarne Helleborine is found on the fixed sand dunes or semi-fixed sand dunes, and these sand dunes are fixed by a species called marram grass and also sand sedge. So it provides a sort of solid base for the orchid to establish, I suppose. So my aims and objective. My aim is to identify the plant communities and soil factors associated with both species of orchids. And to do this, I'll compare the plant communities found next to these orchids and also the soil factors. I'll also look at places where I might expect to see these orchids, but they're not present. So in order to do this, I conducted numerous national vegetation classification surveys, and I'll call it NVC survey from now on. In total, I managed 32 quadrats, and these quadrats are 1 by 1 meter square, and I will identify all the vegetation within the quadrat and give it a percentage cover using the domain value. And in total, 11 quadrats of Marsh Helleborines were quadrated and 11 with Lindisfarne Helleborine. Only 6 without Lindisfarne and 4 without Marsh Helleborine were surveyed, and I'll go on to why that happened. But I'll also compare the Ellenberg values. These values sort of represent species preference for moisture, pH, nitrogen, and salt. I'll go on to that in a bit. So I also did some lab work looking at the 32 soil samples that I took within the quadrats that I surveyed. And using these soil samples, I've conducted NPK analysis, which is looking at the nutrient content. I did loss on ignition to find out how much organic matter is within these soil samples. Basically, I'll weigh the soil samples before and after them going into an oven, and the difference is the amount of organic matter lost through the burning process. Using a probe, I looked at the electrical conductivity and the pH as well. Okay, I'll see if I can articulate this. So the NVC communities are the communities that I ended up classifying for each quadrat. The table fit score is how well the vegetation within the quadrats fit against the classification. I picked out three communities that I thought was particularly interesting. Firstly, the SD17 community is largely made up of silverweed, and this is partly why I didn't quadrat as much quadrats without the Marsh Helleborine. During one of my fieldwork days out with Janet, we noticed that there's a gradient whereby Marsh Helleborine sort of decreased in abundance as the silverweed also increased. Two of the quadrats that I quadrated, I thought there weren't any Marsh Helleborine within the quadrats, and as I was moving the vegetation around, there were two tiny rosettes, so I ended up having to include those into the presence category, so there's a misbalance there. But as you can see, there's still quite a bit of Marsh Helleborine growing in this sort of silverweed community, but SD15 within the presence section is actually also a type of creeping willow community, and I'll talk about creeping willow in just a minute. I want to point out the SD6G community as well, that I would personally associate with the Lindisfarne Helleborine's presence. However, as you can see, SD16 sort of dominates that chart, and that's because there's a sort of problem with the NVC survey in that it doesn't take into account where two communities come together, and so it sort of doesn't classify the area quite accurately, because what I saw in the field is that the creeping willow was actually encroaching onto the marron grass mobile dune community. And if you look at the chart, it says that SD16 is sort of a dune slab community, and that was certainly not the case because the Lindisfarne Helleborine were growing along the dune slope. However, creeping willow, I think, is quite a major species in determining the presence and absence of both species, because orchids are associated with mycorrhizal fungi. And through some research, the Lindisfarne Helleborine and the Marsh Helleborine both utilize a separate species of mycorrhizal fungi, but those two sort of species are also associated with creeping willow. So perhaps the presence of creeping willow can determine whether the orchids are present or absent. And so that's why it's SD15 in the Marsh Helleborine quadrats are also quite important to note, even though I didn't put it in this. So the soil and statistical analysis, to be honest, there's not a lot of that I would feel that it's striking per se, but I did, the pH readings are relatively normal, I'd say, because the sands on Lindisfarne is made up of shells, and they sort of buffer the pH, and so I do expect them to be quite alkaline. In terms of soil, anything above 6.4, you consider it as an alkaline soil rather than an acidic soil. Electrical conductivity, it's as expected again. Quadrats with the Marsh Helleborine and without the Marsh Helleborine are sampled within dune slacks, so you do And because of the moisture, you do expect conductivity to be quite high compared to the dune slopes of the quadrats with Lindisfarne Helleborine and without Lindisfarne Helleborine, because the sand is very porous. It's not going to hold much moisture. Organic content. Within the dune slacks, organic content will be high, and there are a lot more species within the dune slacks, and the turnover rate is quite high, so it allows for organic content to be developed. Again, on the dune slopes, there's hardly any soil content because it's mainly made out of sand, and it's only slightly stabilized because of the marram grass root. Available phosphorus, that's as expected again, because of the organic content within the dune slack. You'd assume there'll be more nutrient in there. But available nitrogen is actually quite surprising. I didn't expect available nitrogen to be higher than the quadrats on the dune slope compared to the dune slacks. I tried to do the test twice to make sure that I got it right, and it's consistent in that the nutrient was quite high in that area. So a likely explanation is that marram grass can have, their roots have some sort of nitrogen fixing properties. I haven't tested that out myself, but it's just through some reading, so that's something that could potentially be checked in the future. Ellenberg values, it doesn't really tell us much. It doesn't separate the quadrats quite well. These values, I got them through looking at all the Ellenberg values of different species found within the quadrats, and I used came up with the mean value to see if the quadrats can be represented, their differences can be represented by the degree in which the plants favor moisture, pH, nitrogen, and salt. There's no difference in that, so it was kind of redundant, unfortunately. So, challenges and limitations. Plant ID was quite a challenge. I haven't looked at the communities within dune systems, and so most of the plants were quite new to me, especially the sedges and grasses. But I had the help of Dr. Janet Simkin and Chris Methrow, who both on 2 different occasions came out to the field with me to help with some ID. Time constraint was a massive factor. Both species flower late towards the season, around June, July time, and that's when I finish all my core modules. So I had to dash out to the dunes straight after to try and quadrat as many as possible. So that sort of explains why some of the quadrats without the Helleborines present were quite lacking compared to those that were present. Or maybe I just focused on the orchids too much, but yeah. I also lost a week due to COVID, but we won't talk about that. I percentage cover, so in order to estimate the percentage cover of the quadrat, I just basically estimate it by eye, and it's quite subjective, which means it's going to be quite hard for somebody else to come along and repeat this experiment and get comparable results, but that's what it is. It is what it is. Ellenberg values, as I said before, they weren't quite representative because they were quite arbitrary. Conclusions and further research. I do feel that plant communities and soil factors, if worked together, can predict the establishment of the Helleborines, but more so the soil factors. In terms of plant communities, I wouldn't necessarily say to use the NVC communities as a be-all and end-all. If you remembered from before, a lot of the Lindisfarne Helleborine were sort of classed as dune slack systems, and clearly that is not the case. But what you can do is pick out specific species, because I didn't actually show you the list of species found within each quadrat, but if you pick out species like the creeping willow, you can probably use that to sort of determine certain areas where you might then conduct soil analysis to then determine whether the soil and the environment is suitable for the orchid. So it's same with a lot of the literature in that Marsh Helleborine are found within moisture-loving parts of the dune and with moisture-loving plants, such as the dune slack. The Lindisfarne Helleborine do coexist with plants that are tolerant of dry conditions, but that doesn't necessarily mean, and that's also a reason why the Ellenberg values are sort of misleading, because some plants can tolerate dry and wet conditions, and that sort of fed into the analysis, which I didn't think was quite a fair comparison. But I think the major factor in determining the niches that these two orchids hold is the hydrology. In studies in the past, conclusions were sort of made in that orchids were separated by ecological gradients, and the hydrology is certainly a main factor in creating this gradient. Hydrology controls the pH, nutrient availability, and lots of soil factors, basically. And I think it's worth investigating how the hydrology shapes these plant communities. Could it be that the hydrology affects the fungi's, the mycorrhizal fungi's ability to colonize certain areas of the habitats? Maybe the mycorrhizal fungi that's associated with Lindisfarne Helleborine thrives a lot more in slightly drier conditions compared to those that the Marsh Helleborine used to germinate. So maybe that's something also that is worth investigating. And the soil factors, the main difference is the organic content, apart from, I think, the hydrology. And that's to be expected due to the amount of plants within the dune slack and their turnover rate, as I mentioned before. So finally, I would like to give my thanks to Dr. Janet Simkin and Dr. Helen Adamson. They were my dissertation supervisor and tutor, and they were really encouraging throughout the course. Fiona McLaughlin, David Early, and Dr. Amy Belf. Without them, I wouldn't be able to navigate the lab. I wouldn't know how to do the chemical analyses and things like that. So big thanks to them. Chris Meffrow for coming out and helping me with Plant ID. James Common for encouraging me to do this talk, but also securing permissions to conduct research at Gossip Nature Reserve for the coral root orchid, but that got scoped out in the early parts of the project. Andrew Craggs from Natural England for granting permission for me to study on Lindisfarne. And my friends from the course, who are also here, they were great lab and library buddies, very encouraging and very supportive throughout my studies. And last but not least, my family for encouraging my change in career and also pursuing this course as well. Thank you very much.